Bismillahirrahmanirrahim. Assalamu alaikum dear second year students. Welcome to your channel The Anatomy Canvas. Today we are going to discuss the histology of the stomach. So stomach learning outcome for today's class is to enumerate the different layers of the stomach wall, describe the gastric glands, differentiate between different types of the glands, and differentiate between fundic and the pyloric mucosa. The stomach is a dilated segment of the digestive tract that digests the food and it secretes the hormone. It receives a bolus of the food from the esophagus, and mixing and partial digestion of the food takes place in the stomach with the help of the gastric secretion, and this combination of the bolus of the food and gastric secretion form the chyme. This chyme is then passed into the small intestine via pyloric part of the stomach. So histologically you can see the three different parts of the stomach. These are the cardiac part, the pyloric part, and the body. So cardiac part is the part of the stomach which is near to esophagus, and pyloric part of the stomach which is near to the pyloric sphincter and connected to the duodenum, and the body or the fundus is the part which is present in between the cardiac and the pyloric parts. So mucosa and submucosa of undistended stomach lie in longitudinally directed folds known as the rugae. You can see in a naked eye examination also the folds are present on the surface of the stomach, and these folds are called the rugae, and the pits are present, which are the indentations in between these rugae. These are called the pits. These are the gastric depressions which are present. So when stomach is filled with the food, these folds flatten out. So purpose of this rugae is to adjust the extra amount of the food into the stomach, not to increase the surface area. So histologically, as other tubular organs of the GIT, the stomach also consists of the four layers. These are the mucosa, the submucosa, the muscularis mucosa, and then the serosa. So mucosa in turn consists of the epithelium, and this epithelium is a simple columnar epithelium formed by the surface mucous cells, and then the lamina propria. This epithelium moves inwards to form the gastric glands, which are called cardiac or fundic glands, present in different parts of the stomach, and then the lamina propria, which is loose connective tissue, and this loose connective tissue contains blood vessels. These contain lymphatic capillaries, this contain the lymphatic nodules, etc., and the parts of the gastric glands. Then this muscularis mucosae is present, which forms a borderline between the mucosa and then the submucosa. So this epithelium, also another important point, this epithelium, when it dips inside, it forms the gastric pits, and gastric pits, the depth of the pits varies in different parts of the stomach, and by looking at the size of the pit you can also differentiate between different parts of the stomach histologically as well. Below the mucosa is the submucosa, and this submucosa is the moderately dense connective tissue, which again containing larger blood vessels, nerves, and lymphatic and submucosal plexus of the nerves. Then is the muscularis externa, which consists of inner circularly arranged layer, outer longitudinally arranged layer, and sometimes an oblique layer is also present, which forms a third layer. And these usually, as the stomach is a globular shape organ, so usually you cannot differentiate easily these three layers, because these three layers intermingle with one another. Outer to the muscularis externa is the serosa, which is again loose connective tissue covered by the mesothelium. So what about the cardiac region of the stomach? First you can see this diagram, that this is the esophagio-stomach, esophagio-pyloric, yes, esophagio-fundic junction. In this area you can see the epithelium is of the esophagus is stratified squamous, and it abruptly change into the simple columnar epithelium of the cardiac region of the stomach. So epithelium lining at cardio-esophageal junctions changes from stratified squamous to simple columnar. Presence of the mucous surface cells and the cardiac glands are present, and then the submucosa consists of Meissner's plexus and the blood vessels. Then the muscularis externa, which is inner circular, middle oblique, and outer longitudinal layer, mesenteric plexus of nerves present in between these two layers, and outer to which is the serosa. So important point to differentiate between different part of the stomach histologically are different features of the mucosa and different types of the glands which are present in different parts of the stomach. So in mucosa of the fundus, you can see that typical gastric glands are present, which are also called fundus gland. The lining epithelium in the fundus is the simple columnar epithelium, and this simple columnar epithelium is formed by the surface mucous cells, and this epithelium dips down inward. Here you can see the pits. The depth of the pit is nearly 1/4 to total depth of the mucosa. It is about 1/4 total depth of mucosa, and then it dips down to form the typical gastric glands. So surface mucous cells are involved in mucus secretion. They contain a large number of mucus in the form of mucus cups which are present in apical part, and because of these mucus cups, the nucleus is pushed towards the bases. You can see in this as well that the nucleus is pushed toward bases. This nucleus is elongated, and along with this nucleus you can also on transmission electron microscopy it is shown that the rough endoplasmic reticulum and Golgi apparatus is also present beneath the mucus cup. So if a proper staining is done with toluidine blue and PAS stains, they show the anionic, strongly anionic group of the glycoprotein and the bicarbonate ions which are present in this mucus. The mucus produced by the surface mucous cell is cloudy in nature. It is insoluble, and it forms a surface, a layer of cloudy mucus on the surface of the stomach, so that prevents it from the effects of the different tough particles, hard particles of the food. This is the transmission electron microscopy of the surface mucous cells. In this you can see surface mucous cells are present, and at some location you can also appreciate the mucus is as present as well, and this is the further higher magnification between the surface mucous cell, and also you can see the mucus is present over here. So the lamina propria consists of the simple tubular, branched tubular glands, and also called the gastric glands, or the fundus glands, or the gastric glands proper. So basically 5 types of the cells are present in typical gastric glands, and these 5 types of cells are: number 1, the mucous neck cells, which are present in middle and lower part of the neck region, and then the in between mucous neck cells, you can see interspersed in between mucous neck cells are the parietal cells, which are also present over here as well. The second type of the cell, the third type of the cells of the chief cells are the present over here. Then another type of the cells are present which are enteroendocrine type of the cells are present in between these chief and parietal cells and the mucous neck cells, and another type of the cell which are fifth type are undifferentiated cells, which are considered to be the stem cells for all of these types. So what about mucous neck cells? These mucous neck cells differ from the surface mucous cell in different respects. First, they have less number of the mucus in their apical part as compared to the surface mucous cell, so that no mucus cups are present over here. Second, their nucleus is usually rounded as compared to elongated basally placed nuclei of the surface mucous cells. Third important point is that the mucus produced by the mucous neck cell is soluble in nature as compared to insoluble cloudy mucus of the surface mucous cells. Another important point about this mucous neck cell is that they are vaguely stimulated. So whenever food enters into the stomach and there is some stimulation by parasympathetic nervous system, so only then these mucous neck cells secrete the mucus, otherwise they are empty as compared to the surface mucous cells, which secrete generally their mucus over here and form a layer on the surface of the stomach. So these mucous neck cells are columnar in shape. They contain mucinogen granules in apical cytoplasm. Nucleus basally located, rounded, and produces the soluble nucleus. The second type of the cell which are very important, these are the parietal or the oxyntic cell present in between the mucous neck cells, usually large, wide or polyhedral, sometimes triangular in shape. These cells having apices facing the lumen of the gland and bases towards the basement membrane, most numerous in upper half the gland, then in the lower part, and they secrete the hydrochloric acid and intrinsic factor. This intrinsic factor combined with vitamin B12 to form complex necessary for the erythrocyte formation. So another important point from your MCQs, that these cells secrete HCl and vitamin B12. Important another point about your MCQs are strongly eosinophilic, large triangular shaped cells usually present in upper half of the gland. Electron microscopy shows that they have a basally located nucleus, and they have a well developed intracellular canalicular system, and also associated with intracellular canalicular system they have a well developed tubulovesicular system as well. Large number of mitochondria are present in these cells which show complex crystal formation. Also the lysosomal granules are sometimes present. They show strong eosinophilia because of this well developed intracellular canalicular system. Rough endoplasmic reticulum is present in basal areas, and these cells secrete hydrogen ions and chloride and also the intrinsic factors as well. So this is an H and E stain section of the typical gastric glands, in which you can see these large eosinophilic cells. These are parietal cells which are present over here, and in between parietal cells you can also appreciate the chief cells are present as well. These chief cells are also called the zymogenic cells. So this is a little bit diagrammatic presentation. You can see the chief cells, the parietal cells present over here in this diagram as well. This is the lamina propria and then the muscularis mucosae. So what about chief cells? The chief cells located in lower third, usually lower third and most of the time at the bases of the glands. They contain the rough endoplasmic reticulum near base, secretory granules near apex, and small Golgi apparatus secrete pepsinogen, which is converted into pepsin in an acidic environment. This is the electron transmission electron micrographic picture of the chief cells. You can see basally located nucleus and the basally located rough endoplasmic reticulum, which gives basophilia to these cells. Also in the apical region you can see the zymogenic granules. These zymogenic granules stain with eosin. So in apical area you can also appreciate the eosinophilia is present, but at most of the time these zymogen granules are not well preserved. So usually these cells appear basophilic because of a well developed rough endoplasmic reticulum in basal area, and also because of the large nucleus which is present over here. This is the H&E stain section, in which you can appreciate very well the parietal cells. These are the large triangular shaped eosinophilic cells, and interspread in between these two cells are the chief cells. Also the mucous neck cells are present over here. Another important point, students, you must remember that these are not the pink cells. So please correct yourself. These cells do not stain pink. They actually stain with eosin and show eosinophilia. Similarly, these cells do not stain with blue pencil. This blue pencil is actually hematoxylin. Most of you students have a mistake. You do not know the names. So this is hematoxylin, which show basophilia in this chief cells. So please correct your terminologies. The chief cells stain with hematoxylin, and the parietal cells stain with the eosin and show eosinophilia, and chief cells show basophilia. Also the mucous cells also show small amount of basophilia because of mucinogen, and present in the cells also stain with the hematoxylin. Another slide in which you can see the larger cells which are present are the parietal cells, and in between parietal cells you can see these cells which are chief cells are zymogenic cells. Another type of cells, enteroendocrine cells, also called APUD cells. This is another important MCQ, and you people do not know what is APUD cell. This is actually amine uptake precursor and decarboxylation cells. These are large number of the cells which are present throughout the gland, but you cannot appreciate these cells, you cannot identify these cells in typical H&E stain section. These are small cells usually located at the bases, sometimes do not reach up to the lumen, and these cells secrete serotonin, histamine, gastrin, and many other factors. They are one of the cells of the cell lineage of the enteroendocrine system of the GI mucosa. And the last type of the cells which are present in the typical gastric glands are the undifferentiated cells, which are also considered stem cells, and you also do not see these, do not differentiate these undifferentiated cells from in the H&E stain section especially. So here we end our cells, and now we move on to muscularis mucosae. Consists of the two thin layer of the smooth muscle, outer longitudinal and inner circular, and then is the submucosa, which contains the lymphatics, blood vessels, and meso nerve plexus. Then muscularis externa consists of oblique, circular, and longitudinal, and then the outermost layer, which is loose connective tissue covered by the mesothelium. Here we end our fundic glands or fundus and body of the stomach. And this is another slide very well showing the typical gastric gland. You can see the depth of the pits over here. These lines show depth of the pit, and this is the whole thickness of the mucosa, and you can well differentiate that this is just 1/4 the total thickness of mucosa. These pits are total 1/4 of total thickness of mucosa. This is one of your identification point on slides for histology or histological slide for identification of the fundus. And second very important point which you must understand for to identify your slide is the presence of the parietal cells, which are large eosinophilic triangular shaped cells present in typical gastric glands. These are not present in pyloric or the cardiac glands. And here is the submucosa, then the here is muscularis mucosae, then the submucosa, and then the muscularis mucosa over here. Then is the pylorus. Very few points to differentiate the pylorus from the first shape of the glands. These are branched tubulo-alveolar glands and usually tortuous. Second important point is pits. The pits are nearly half the total thickness of the mucosa. Pits are very deep. Third important point, the surface mucous cells and mucous neck cells are present in the and along with the enteroendocrine and undifferentiated cells. There are no chief cells and there are no parietal cells over here. Rest of the gland are same with the fundus. Muscularis mucosa shows same feature as the fundus and also the differences between the three parts. Cardiac part contain cardiac gland, fundus contain gastric glands, and pylorus contain pyloric gland. The pits are less deeper, nearly 1/3 total circumference of the mucosa. Pits are less deeper in fundus, 1/4 total thickness of mucosa. Pits are more deeper, nearly half or 2/3 thickness of the mucosa. The parietal cells are absent, and if present, they are very few. Some more parietal cells are present, and parietal cells usually also absent. If present, they are very few in number. So different types of cells, and you should memorize this table according to your MCQ point of view. The parietal cells secrete HCl, intrinsic factor. Function is to kill microbe, activate the pepsinogens, and vitamin B. Intrinsic factor binds to vitamin B12 to allow its absorption. Then chief cells secrete pepsinogen for protein digestion and gastrin lipases for fat digestion. Then G cells secrete the gastrin, stimulate gastric acid secretion. ECL secrete the histamine, stimulate the gastric acid secretion. Mucous neck cells secrete mucus and bicarbonate, and this bicarbonate actually neutralize the HCl secretion of the lumen to prevent prevent the effect of HCl, its ulcerative effects from the epithelium, and protect the epithelium. And D cells, these all the cells, G cells, E cells, and D cells, they are the different types of cells of enteroendocrine population of the cell. They secrete somatostatin and inhibit the gastric acid secretion. So dear students, these are your references: the basic histology by Lycosan Siddiq, the histology text and atlas by Jan Quera, and histology text and atlas by H. Ross. Here I complete my lecture, and we will inshallah meet in the next class with the histology of the small intestine. Till then, dear students, Allah Hafiz.